All right, good morning, everybody. How are y'all? Good? I'm pretty good. So, y'all are very kind. My first class of the day, part of it's that they're ninth graders, but part of it's the first class of the day. I'm like, morning, and they just look at me, be dead silent, and then after I stare at them for a while, somebody will be like, morning. Those are the same ones that, like, as they walk in by the door, I'll be like, hey, how's it going? And they don't even look at you. So, all right. For our AP classroom, let me zoom in on this a little bit. If you're wanting to get into AP classroom, which you should, these are our join codes. So you might have already started signing up in some of your classes. You can sign up right now if you're on your phone or take a picture and use it later. I will copy paste for those of you all on Zoom. Let's see.
So are we second or are we third period?
We are third period. Yes? That's all right. It's fine. Yeah, no matter which class you sign into, whether you sign into second period or third or fifth or zero or whatever, that's just for us, for our organization. College Board doesn't care. And this year, so many people are going to be switching around and stuff, it doesn't matter too much. You're welcome. So for you guys, I'll put it in the copy. Put it in the chat. There you go. If you want to go to AP Classroom, this is how you access some AP stuff. It's also how you're able to sign up and buy tests for AP exams in May. And we're in third period. Actually, I'll put you guys on the 0 period code. If you already signed into third, that's fine. So 0 period code. Let's see, that is, looks like you can see it on the screen, but just in case, 7ENW4. So we'll call you guys 0. Again, if you sign into third period, that's fine. All right. So from there, you can buy AP exams. Does anybody know when the $5 discount goes away? October 2nd. So that's like Friday. Cool. I don't know how much exams are. They're usually 90-something. Does anybody know how much they cost? 95? So if they're 95, then if you sign up by Friday, they usually have a $5 discount per test. It's not a lot, but if you're taking 3 or 4 or 5 tests, it's $15, $20, $25. So if they're currently 95, then if you sign up by Friday, they're only 90. As far as canceling tests later on, the deal that was in place before all the COVID crap hit last year was, I think you got about half your money back. Like, I think if you sign up now and then in like April you're like, yeah, I don't want to take it, I think maybe you get $40 back or 45 or something. So you get about half your money back, give or take a little bit. Yes. So the thing that sucks about signing up now is, like, you've had one test, so you don't have a whole lot of data to go on. You probably don't have a very good idea. I don't have a very good idea of, like, whether I think you get a 5, 4, 3, 2, 1 yet, just because I only have one test and one quiz for you. So it's a little bit tricky. I will say that overall, students tend to be pleasantly surprised. So students that take it and they're like, yeah, I'm not going to do that well, I think I'll get a 2. A lot of those students get 3s. There's always surprises down, but there are more surprises up for students. So if you're kind of on the fence and you're like, it's $95 and you're going to school next year, 95 is nothing in the world of college. Like we're currently using the first edition of this book, so I think Lacey said they're like $125. They're on the 3rd edition. So if you get like the latest, the 3rd edition, it's like 200 something. So you'll sign up for bio class, and that's thousands, and then you like go to buy the book, and it's like, the book's like 200. Oh, then you need the lab manual. That's like 80. And then you need this, and you're like, crap, I haven't even done anything. Like, I've spent $1,200 on books. So all of a sudden AP exams look super cheap. So yeah, I'm happy to try to give you some guidance along the way as to whether or not I think you should sign up or what I think you'd do. But again, I've got one test and one quiz, so it's my best guess. All right, any questions about AP Classroom stuff? Okay, now, it doesn't have to be everybody, but if a few of you guys would try, we might have an electronic textbook. So if you go into K-Cloud, I'm going to pull this over so you can kind of see what it looks like, and you all at home can try this also if you want. So let me drag that over. So my K-Cloud is obviously going to probably be littered with a lot more crap than yours because I'm a teacher. But somewhere on your K-Cloud, I want to see if it says Bedford, Freeman, and Worth. That's like our textbook manufacturer. So they do have a digital book, and Keller has access. But of course, this year is the year that we can't access it. There's all these problems. Years past, you just put in a code and you're good to go. But this year, nothing works. Okay, so if I click on Bedford, Freeman, and Worth, for me, I get in. So I can see that I have the textbook if I can find my mouse. Oh, wait, the screen share stopped. Hang on. That's fine. I'll just keep it on that camera. Okay, so let me bring that back over. So Bedford, Freeman, and Worth. And then when I go in there, of course, it's got like a billion AP Bio classes for me, and I just click Go, and I'm good to go. Do you guys see anything like that? You do? Okay, this is promising. Then you get here. Yours, you might have to it says, does it say enter your course? Oh. My fingers are crossed. This might be me thumbing my nose at 2020. When you click on that, does it give you like chapter 1, chapter 2, chapter 3? We have a digital check. Yeah. Yours has a problem? How many of y'all can see like chapter 1, 2, 3? 1, 2. Okay. What does yours say? Uh-huh.
And it tells me to pick a course, so I pick it, and then it does this.
Well, damn it. Okay. Do you have a paper copy of the book?
Is it the book we got from?
Mm-hmm. Yeah, that we got like a week ago. Do you like using the paper copy?
I don't mind it.
Okay, cool.
Do you need to bring it to school every day?
Oh, no, you don't need to bring it to school. It's the only time we use the book is like at home. So I was just making sure that you have access. How many of y'all, yours has a tech issue? So some of you guys, everything's cool, and some of you guys are like, we're sorry, there's a technical problem. All right, perfect. Well, all of you guys in person have a paper book. Those of you all over here, let's see what it says for you guys. Go to take your test. Yes. Okay. All right, give me like 2 minutes and I'll be right back. Okay. So for your test, let's see. Okay, we're going to take like a 3-minute break. second, home crew. Got to get a little bit of test code related stuff done. All right. So for those of y'all that are having trouble with tech, I might have you try it again later, but I'll see if I can get it fixed. I don't know. Took them 6 weeks to get it going the first time. I'm not sure how long it's going to be before it gets fixed. But in there, if you're able to get in, chapter 1, 2, 3, 4, it's just a digital version of our textbook. If I were you, even if I was remote, I would contact odalis.smith@kellerisd.net and ask for an AP Biology textbook. So, all right, let's see. I think we're ready to actually start class now. So, it's good to see you all. If for some reason we get quarantined, I don't know anything that you guys don't. I just know that more and more students are getting quarantined. If we get quarantined, you'll get emails that come from Keller ISD. They won't look like they come from Allen Cotton. And your email filters might actually grab them. So just make sure you're checking junk folders and stuff. So if we got quarantined tomorrow and like a day or two goes by and you're like, none of my teachers have contacted me, check your junk mail, right? Like certainly somebody's going to contact you within probably a few hours. Okay, so today we've kind of got two things that we're working on. We've got a little bit of stuff to learn like biology-wise, and we also want to prep for our lab. So if I pull this down just a little bit. So today we'll do our pre-lab, and we'll talk about Chapter 5, Section 5. Tomorrow we'll be in lab. I'll take you guys in there remotely. I'm not sure exactly how that'll go. I might try it and then find out that I just need to like record some video and you guys watch it later. I'm not real sure because my cords are only so long. So I'm going to try to take you guys in there with us. And so tomorrow we'll be in lab. So we pre-lab today and then like, not tomorrow, but next class. So on Thursday when I see you guys, we'll be across the hall in lab and you can walk in and you can go ahead and get started. You don't have to wait for me to say go. You'll have all the information you need after today. So we'll pre-lab, do 5.5, and then lab here. In the gradebook, labs count half as much as tests. They're both summative, but I'll weight your test twice and I'll weight labs once. But they do count as summative, so it is a way to kind of bolster test grades a little bit if you need that. Okay, so I've got to hand out some stuff. You'll get two things. And for the Zoom crew, I'll show you what they look like. They're in your Google Drive. But I'll show you what they look like. You're welcome. Just so you know you're getting the right thing. There you go. You're welcome. There you go. There you go. You're welcome. You're welcome. You're welcome. 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 You're welcome.
What do I do if I'm not going to be here?
So I will like record little clips of like, here's the setup. 5 minutes in, I'll go over and have like a 1 minute clip. So there will be almost like a, I don't know, you know how most labs, like you set it up and then you like wait for 5 minutes and take a reading or wait. So I'll kind of keep coming back every few minutes and have video, and I'll put it in our Google Drive. So before you leave today, if you'll remind me, I'll put our Google Drive up there so that you can get to it. Yeah, it kind of sucks. You're going to miss lab day. Like this lab, I like all the AP Bio labs, but this isn't like one of the top two. The DNA labs are the best ones. So those are the two that you really don't want to miss. And here's one other page for you guys. You're welcome. There you go. 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 You're welcome. You're welcome. No problem. You're welcome. Welcome, welcome. You're welcome. All right. So you have two things, and in the Google Drive under AP Biology lab, there'll be two things. I might have called it like Lab 1 or Osmosis Diffusion Lab or something like that. And you'll have one that looks like this. Let me switch cameras. One of them that looks like this in there. It says Lab 1, Diffusion and Osmosis. We're basically doing two labs at once. And then you'll have like this thing that I call like the Lab 1 page. Looks like that. And on the back. Okay, so the way that labs work in here is you'll pretty much always get two handouts. And the like front and back thing with the questions, that's what you actually turn in. So this is for you, but you don't turn this in. Sometimes, like in this lab, I'll have you like cut out a data table. Like if you look on the very back of this, it literally says like you're going to cut this out and paste this on your one-pager. Like it handwritten out to the side, it says you will cut out this data table and attach the back and top to your one-pager. And on my one-pager, it even says, staple it right here. Like there's a little X. It says you're going to staple it right there. So on the day whenever you turn in your lab, which is for you guys the 7th, whenever you turn in your lab, you'll just turn it in like this. You'll have your data table stapled to it, and I don't need the rest of this. Sound good? Okay. Whenever you do labs, of course you'll be in groups, and we'll have hand sanitizer and stuff like that. We'll be, kind of aware of like, if I'm running this part of the lab and I'm touching stuff, then probably the other people at my table shouldn't also be touching the same stuff. So we'll be aware of that because of COVID-19. But you can work as a group to get your answers, but you should be like talking about them instead of like copying them. So we might be working through like, hey, number 1 or number 2, what do you think? And we kind of talk about it, and then we'd like put it in our own handwriting. You don't want it to look like somebody wrote it and then somebody else just sat there and copied it. Some stuff, like number 3 there, where you're just putting things in order, they are going to probably be identical to everybody in your lab group. Like it's a numbered list, and things are in a certain order, and that's kind of it. On the back, where it has the graphing paper, you're going to build a graph. Don't forget 7th grade graphing. Don't forget titles and labeling axes and scale, all that crap that you've learned 10 different times. I don't reteach it. But I do tag you for points if you don't do it. Okay, so for the lab, for the pre-lab, I'm going to kind of walk you through how everything's going to go down. So I'm going to try this. And I might have you two switch because I'm gonna be teaching on that board, I think. So if you two will just like kind of move over here for right now. Thank you. Move all my hot plates and stuff. Okay, let's see how we're looking here. It's a close-up of my face. All right. Okay, that's the warm one. Then I need this one. Okay, so if you think back to your 9th grade class, most of the 9th grade classes did a lab where you tested for like carbohydrates and starch and lipids and stuff like that. It had been one of the first labs that you did, and there were all these like kind of color changes and stuff. So we're gonna actually use some of that stuff from 9th grade. I'll still re-explain it because I know 9th grade's been a while. So let's say that I've got some starch, so just regular spray starch. You can use it on clothes to like make your shirts look crisp or whatever. If you're in like military, you have to starch your clothes, for example. And I've just got a little bit of water from my sink over here. By the way, you can't buy spray starch in stores right now. They're out. I don't know why. I don't know if the people are hoarding it for COVID. Like, I don't really know what's going on. There's a pandemic. We need spray starch. Go get it, man. So I put a little bit of that in there. Add a little bit more water. So the starch test is actually really sensitive. I didn't put very much in there. Make sure you guys can actually see. Yes, you can. Warm one and other one. Okay, so I've got my spray starch water in here. And let's see, got a stir bar. Here it is. This little stir bar, some of you guys have seen this, some haven't. But our burners, they've got 2 little dials. One of them is for heat. That's, the burner function. And another one is a stir bar. So this guy right here, I've got a little magnetic bar. I can drop it in there, and I can turn on the stir feature, and it'll start stirring that around. I'm going to add just a little bit more water to it. Hang on. All right, that'll be a good effect. So it can stir it. There's a lab that we used to do in AP Bio where I had to dissolve potassium permanganate. And it would say, mix this much potassium permanganate with distilled water and then stir it for 8 hours. And you're like, I don't wanna. And so what you do is you just put a stir bar in here and turn it on, and it'll stir for you. So you can't tell yet, but it's spinning around and stirring. So I've got some starch water stirring, and then I have some iodine. So this is Lugol's iodine. And if any of y'all have ever had like a IV, maybe you had anesthesia for surgery, and they rubbed some like yellow stuff on your hand before they stuck you with an IV, that was Lugol's iodine. So it's an antiseptic. We can rub it on your skin. We do that at a hospital, but it does stain. So it's kind of this brown color. So what we're going to do is add some of that to starch water. We'll see what happens. So as I start to add it, it turns kind of like a dark bluish purple color. And eventually, it'll turn so dark that it looks black. almost like food coloring. Glucose, whatever. So I'll add a little bit more to darken it up some. And this is what a positive starch test looks like. So if I didn't have starch, then, and I added this kind of brownish iodine colored stuff, it would just be kind of like dilute brown. It'd look kind of like, I don't know, like this, only dilute. Okay, so now, one thing I can do just for fun, this has nothing to do with the lab, but I can oxidize this and make it go back with hydrogen peroxide. I can just go back and forth. This is what I do on the weekends. Just come up here and mix stuff. There's still enough hydrogen peroxide to react and take it away. So it just kind of keeps on going. And eventually, if I put in enough of the iodine, then it'll eat up all the hydrogen peroxide, for lack of a better word, and it will go back to purple. So this is a positive test for starch. Okay, so what we're going to be doing in our lab is we're going to be using some dialysis tubing. So what is dialysis? Or let me ask it this way. What organ is failing you if you're on dialysis at a hospital or something? Yeah, your kidneys. So dialysis is basically, if your kidneys are failing, then we can do dialysis, and that can kind of take the place of your kidneys for a little while. So the first lab that we do is called 1A, and it's a diffusion lab. So what we're going to do is we're going to use some dialysis tubing. Let's see. So your dialysis tubing will be soaking in water, but it just sort of looks like clear tape. If I can get it just like clear tape, it's hard to find the edge. There you go. So it just looks like clear tape like that. And it doesn't seem like it's hollow, but if I dip it in water, let it soak for a little bit, then what I'll find is that if I kind of like rub the edges like this, just back and forth, kind of harshly, it's actually hollow. This is a hollow tube. If I had like a funnel or something, you can see I've like got the funnel in the tube. So this is now a tube. And it's dialysis tubing though. It's not like Ziploc bag or something. So it has very specific sized, teeny tiny microscopic holes in there. So somehow a machine extruded this plastic, and then they punctured holes that were like however many nanometers in diameter. So this has like millions of teeny tiny little bitty holes in it. And if you're in the hospital, we can filter your blood through it, and that's similar to how your kidneys work. So a little bit more complicated than that. But so now I can use this dialysis tubing as a semi-permeable membrane. Picture like a cell membrane with channels. This is now a cell membrane with channels. So what I'll do is I can like tie off one end of this. Just use a knot. Tie it off on the end, and I want my knot kind of near the end. I want it like overlapping enough to where it's not going to leak. But I don't want to put it like right in the middle of my dialysis tubing. And now if I wanted to, couldn't I like take a funnel, put it in there, and then fill this with liquid? I could. So that's what we're going to do. So for part one, I'm going to fill my dialysis tubing. This is my dialysis tubing. There'll be a glucose and starch solution. And then I'm going to put that in a beaker. But we're not going to use beakers because I end up needing like 80 beakers. And so we just have some plastic cups, and you'll have a Sharpie so you can remark them. These have been used a lot. So I'll basically have a beaker full of water, and inside of this I'll have a glucose starch solution. I'll tie off the top, and it's just going to float in there. Into the beaker solution, I add some Lugol's iodine. That was the reddish brown stuff that we just added. Okay, I'm studying diffusion. So why is this a logical setup to study diffusion? Like, why does it make sense that this could be a diffusion lab?
If it replicates the cell membrane, then when you put the glucose iodine in it, it will diffuse into whatever substance you put it into.
Yeah, so I've got several substances. If you couldn't hear, she basically said if it's replicating the cell membrane, if I put it in iodine, like, what would happen if starch and iodine met? How would I be able to tell? Yeah, it turned purple, right? If they didn't meet, it wouldn't be purple. So my glucose starch solution is going to look, just like regular water. Whenever I take my beaker water and I add iodine to it, it's going to look like iodine water. So it'll look like somebody that used the restroom, but they're dehydrated. So my beaker water is just going to look like kind of rust-colored iodine water or apple juice or something. And then this will be floating in there. And hopefully I can see, like, does iodine move in or does starch move out? What about glucose? We've got to have a test for glucose. So we've got a glucose test also. For the glucose test, we do this thing called the Benedict's test. And let me get that set up. So here's some glucose. It's dextrose. That's right-handed glucose. Tastes better. It's a carbohydrate joke. It's the non-funny kind of joke. I specialize in the non-funny kinds of jokes. In fact, I would say I'm the best at them. So I put in precisely that much. Mix it well. Okay, so here's some glucose water. Now, to do a test on glucose water, to see a positive for glucose, I use this stuff called Benedict's solution. Sorry, I was originally going to do all this up there, but I'm slowly moving my demo over to here. So I've got some sugar water, and I want to somehow see that I have sugar water. So I've got to figure out a test that tests for sugar, for glucose. So I have this stuff called Benedict's solution. It's a really cool blue. It's copper-based. It's got copper oxide. When copper meets oxygen, it turns blue like the Statue of Liberty. The Statue of Liberty is actually coated in copper. You might have had a penny at some point that you found that kind of turned this color blue. Maybe not, I don't know. Okay, so the way that I get a positive test for glucose is, this is my glucose water. I add some Benedict's solution. And at first, it just looks like basically what you'd think. It looks like blue. If Benedict's is blue and then I mix it with sugar water, it just looks like dilute blue. So what I have to do to get a positive is I have to put it on the heat. So I'm going to heat it up. Now that's going to take a minute. So far, does this lab make sense? Okay. If glucose enters or exits or whatever, how would I be able to test for the presence of glucose? With this glucose test. If starch or iodine's moving in and out, I'll be able to test by seeing a color change. Right? Like if this is my iodine water and some starch water mixes with it, then it turns dark. Okay, so how would I test for glucose? I'll do this glucose test. So any color change in this represents a positive test. So my water's hot. It won't work without heat, so I'm kind of heating it up in a water bath like this. So I'm going to leave that for the moment, and I'm going to tell you about the osmosis lab. For the osmosis lab, what is osmosis again? Yeah, water diffusion. When water's moving from high to low, that's osmosis. So you'll have 6 more of these cups, 1, 2, 3, 4, 5, 6, and they're all going to be filled with water. That's H2O. And I know that you know water's H2O, but if you're looking on the camera and you can't quite read that, that says H2O. I am aware that you guys know that water is H2O. So I'll have all these cups, and now what I'm basically going to do is I'm going to take dialysis tubing, and I'm going to fill this one with 0.0 molar sucrose solution. Sucrose is table sugar. Like I literally just have this giant thing of sugar that I got from Kroger to mix this stuff up. So what is molarity again? Like what am I measuring? Like if I'm measuring time, I might use seconds or minutes. What's molarity? Yeah, like concentration. So how acidic is this acid, for example? By the way, we're starting to see a color change. So if I look at this, it's kind of starting to go yellow. Let me give you a white background. Kind of starting to turn yellow. If I leave it in there a little bit longer, it goes from greenish to yellowish to like University of Texas orange. So it should go pretty quickly now so we can watch it. Just kicking back, watching Benedict's solution do its thing. Send. Okay, so again, it's already testing positive for glucose, but the more glucose it has, the more kind of orange it'll get. I'm going to come back to that. It's going slightly slower than I thought it was going to. Normally my water is hot enough to where like it would take maybe another 20 seconds. It'd make it through the yellows, greens, and into the oranges. All right, so, oh, yes.
I have a quick question. How do we tell if the starch was going into the iodine? Like, would there also be a color change, or would we test it with the heat water with glucose?
Okay, so you would basically want to check, like if I was over here and I wanted to test this liquid, I'll have to test it twice. So one test would be the starch test, and then I would pull some of this liquid out. If I wanted to see if there was glucose out here, I pull a sample of glucose out, and I run this test. It's starting to orange up a little bit. And if I wanted to test the inside for glucose, I could pull some out of there. So I can basically pull some liquid out and do a starch test. I can pull some liquid out and do a glucose test. Does that kind of make sense? So that would be a no. So which thing were you, well, no, it's okay. So which one were you wanting to test, for example? The fluid that's in the beaker or the fluid that's in the dialysis tube?
The fluid that's in the beaker.
Okay, so the fluid that's in the beaker, how would I know if, well, first of all, is iodine going to be out here? Yes, because I put it in there. So if starch leaks out of the bag, starch starts in the bag. If starch leaks out of the bag, how would I be able to tell?
That's my question.
Okay, what happens when starch water is clear? Lugol's iodine water is kind of rusty colored. What happens when starch and iodine meet? Yeah, it turns black. So if starch was leaking out, where would it turn black? Yeah, in the cup. What if instead it turned black on the inside? What does that mean? Iodine went in. So it depends on, black means that starch and iodine met. It's just, where did that actually happen? All right, and then this is a full positive test for glucose. So it's like University of Texas orange kind of colored. All right. So now for the osmosis one, molarity measures concentration. So I've got water in this beaker, and then inside the dialysis tubing, I've got 0.0 molar sucrose. So that's water with water. Over here, I've got 0.2 molar sucrose, 0.4, 0.6, 0.8, and 1.0. So which one's the most concentrated? 1.0. So this is the sugariest of the sugar water. In fact, this is so sugary that I have to boil the water to get the sugar to mix. And if I just put the sugar in the bottom of a giant graduated cylinder and pour boiling water on it, it won't mix. You have to like add water, add sugar, swirl, put it in the microwave, add water, add sugar. It's like yellow. It's so full of sugar. So this is like the sugariest sugar you've seen. It's almost like corn syrup sugar. And then what we're going to do is we want to see if osmosis is happening. So the way that we'll do it is we'll weigh these things. So we got some of these little bitty scales. You can buy the big ones that you have in chemistry for $100, or you can buy these for $8. And they both measure to the same level of precision. They both go 1 digit to the right. So we've got these. And what I can do is I can weigh my dialysis tubing before I put it in here, and then weigh it after, see if it gains or loses weight. So whenever I fill this up, here's my dialysis tube. I'll fill this one up with like 0.4 molar sucrose, or maybe I'm filling it up with 0.8 molar sucrose. Then I can weigh it. Anytime you weigh something on a scale, you want to use what they call a weigh boat. Do you guys use these in chemistry? So I'll have a weigh boat on there, and so here's my weigh boat, and I'll put my dialysis tubing on there, and I can weigh it. I'll do the experiment. I'll put it in here. If it was this one, I'll put it underwater. The lab's going to tell me to keep it under there for like 20, 25 minutes, something like that. Then I can take it out and weigh it again and see if it's gaining weight or losing weight, and see if that helps me explain some osmosis stuff. Make sense? Okay, so in biology, the tough thing about AP Bio labs is usually timing. The lab itself isn't that hard, but it'll be like, all right, get this set up and then wait for half an hour. So if you get into lab and you don't get going right away, all of a sudden you're like, oh, this isn't too bad. And then like the next step is wait 30 minutes and you're like, crap, I'm going to run out of time. So you kind of want to like have a little bit of a game plan before you go in there. That's why we pre-lab today for a lab that we have on Thursday. So you guys are familiar with what our labs look like. There's 7 lab stations in there. You can pick who you want to be with. 3 to 4 per lab. For a class this size, we can probably 8, 9, 10, 18, 19. So we could probably have 3 per lab in there. And basically, once you get in there, you can start like setting up dialysis tubing, getting your cups, and all that stuff. So when you come into class, at the front table, I'll just have like all the cups. And you'll have your dialysis tubing. You'll just come up there, and it'll be very obvious. There'll be 7 rows, and you just like grab everything in the row, and you can get started on your lab. Does that make sense? I know until you're there, you don't exactly know what everything looks like, but that's what we're working on. So between now and next class, you don't have to memorize the lab or anything, but look through the procedure and see like, all right, what things can I do like the second I get in there? I need to get a dialysis tube and fill one of them with glucose starch. And then we also need a dialysis tubing filled with this, and this, and this, and this, and this, and this. So if there's 3 of us, we can get started right away. So it's like, all right, you and me, I'm filling, or me, I'm filling these dialysis tubes. And then Bob over here, he fills these 2. And Jane, she fills these 3. That's the part that takes the most time, is filling dialysis tubes. So go ahead, and as soon as you get in, get rolling on that. Every lab station is going to have, 2 or 3 funnels, so you could have 2 or 3 people working simultaneously. Sound good?
So will we have to soak the dialysis tubes in water and then do all of that?
They're already going to be soaking for you. So basically anything to get the lab ready to make it to where you guys can just kind of pick up and run. So yeah, but everything will be there. Like you'll have a stack of 1, 2, 3, 4, 5, 6, 7 cups. You'll have a cup like this to where there's already dialysis tubes soaking in there, and you're just ready to go. So you'll walk in, and the first thing you'll do is put out your lab so you can kind of read it, but then you'll be like, all right, tie a knot in one end and start trying to open these up. That takes a while. I make it look easy, but I'm a professional. So that's why they pay me the non-big bucks. All right, feeling okay on lab? Cool. So now let's switch back to our other view.
I do have a question.
Yes.
For the osmosis ones, do we weigh them first before we soak them in, and then
Yes, because we're wanting to see if they gain or lose mass. So this one, before I throw it in the water, I'll go over and I'll weigh it. Everybody will have their own scale. So at your lab station, there'll be a scale. So if you want to weigh this one, write down the weight. Weigh this one, write down the weight, that type of thing. Because you're going to have all these different cups, you can see that we've been using them for years. You'll have a Sharpie so you can mark through and label them. That way you can keep track of what's what, because otherwise they all look the same. So I'll have one cup and I'll label it 0.4 molar. I'll have another one that's 0.2 molar, something like that. All right. Switch this around. Okay. I don't see anything in the chat. And you guys at home won't have as many questions about the lab because you're just going to be kind of observing it. You don't get to do it. Sorry. Unless you have some dialysis tubing at home. I don't know if any of y'all are on dialysis. Just steal a little bit of tubing out of there.
Oh,
Huh?
also, so it says that you'll cut out this graph and put it on your one-pager. Is this your one-pager? Or is it
Yeah.
separate?
Yeah, so the one pager is this like front and back thing, and that's all you turn in. So every single lab will have a front back one pager, and that's all you have to turn in. Sometimes I'll have you staple a data table to it, but it'll be like that's all you turn in. So yeah, this is our one pager. Good question. Any others? Yeah.
And this is like, this whole thing is to do during the lab, not like anything before?
Yeah, none of it you have to do before. So as far as before the lab, it just kind of like look through the procedure part, just so you kind of have a plan. I don't have to have it memorized, but I want to be able to have like the second I get in the door and I get my supplies, what's the first thing I can do? And then while I'm doing that, I can like figure out the next thing I'm going to do, just for efficiency, just for timeliness. So there's no pre-lab questions you have to fill out ahead of time. I know some teachers will make you write out a procedure or answer some questions. I don't. So you don't have to do or turn in anything before lab. It's mainly just when you get there on Thursday and the rest of your lab partners are ready to go, you don't want to be that kid that's like, what do we do? And they're like, shut up, get out of the way, you're slowing us down. Like, why don't you come prepared? That kind of thing. Any other questions? Cool. All right. So the thing that we're looking at next, if you are following along in your notes, is down at the very bottom of page 2. So page 2 of your notes, down at the very bottom it says concept 5.5. Right below this like group of pictures. 5.5. Okay, so it says concept 5.5, a membrane plays a key role in a cell's response to environmental signals. The stuff we get to learn today is really cool. As I always say as a disclaimer, as a bio teacher, I think it's all cool. But the stuff that we're getting ready to do, students also think is cool. So let me grab these pictures and pull them over. All right, so we'll look at that picture a little bit more closely in a second, if you're remote. But under these 4 primary types of cell signaling, we got the first one. What do I do first? Juxtacrine or direct contact. So direct contact, this is ways that cells can talk to each other. So cell communication is huge. If my adrenal glands release adrenaline, it has to go into my bloodstream, eventually make it up to my brain, hit a receptor in my brain, and then my brain says, speed up your heart rate. Diaphragm, speed up your breathing rate. Stuff like that. So this cell is basically, or these cells are telling these cells to tell these cells to like move faster. So cells are always talking to each other. If I get a cut, like right there, and all those cells die, well then the cells on either side have to grow, and they have to grow until it's time to stop growing to fill the gap. And if they keep growing, that's cancer. And if they don't, I have a giant gash there forever. So cells always have to talk to each other. So the first way that cells can talk to each other is juxtacrine, or direct contact. Direct contact. So a cool example of direct contact is the heart. So let's say these are heart cells. And I've got a channel right there in that heart cell. All right, so it turns out that heart cells contract on their own. So they need oxygen, they need ATP, stuff like that, but your brain doesn't have to tell your heart cells to contract. They contract on their own. Now, your brain can take over and tell them to speed up, but if we disconnected your brain from your heart and we kept your heart cells alive, again, ATP, oxygen, pulled off CO2, they would actually keep contracting. So how does that actually work? Well, we look at muscle contraction later on in the year, but the quick version is whenever calcium goes into muscle cells, they contract. Okay, so that's something you just, for the moment, you just have to accept. Later on in the year, we'll see how, but when calcium enters a muscle cell, it will contract. If I could take a syringe and go into muscle cells in my arm and just squirt calcium in there, they would just automatically contract. That is the first chemical thing that makes contraction happen. So I've got direct contact. Here's a heart cell. It's got this little channel. Well, the way that your heart beats is actually pretty amazing because calcium just leaks in. So if I've got calcium out here, here's calcium Ca2+. There's a bunch of calcium outside, not much inside. Once enough calcium leaks into a heart cell, that heart cell contracts. So I'm just going to make up numbers. Let's say that it's 100. Once 100 calciums make it in there, then that heart cell contracts. So here's my heart cell, calcium Ca2+, and 100. So 2, 4, 6, 8, 10, 98, 99, 100. At 100, this heart cell contracts. But then my heart has to all those cells have to contract together in a coordinated manner. Otherwise, you have fibrillation, which is like this. Here's your heart. If they're just all beating to their own pace, then your heart does this. They compare it to a bag of writhing worms. So sometimes people's heart just kind of gets locked like this, and it's just going like that. It's not pumping blood anywhere. We need it to be like dun dun, dun dun, dun dun, dun dun. Atria, ventricles, dun dun, dun dun. The sound is the valves closing. So bum bum, bum bum. So they have to work together. Okay. So have you guys heard of the pacemaker of the heart? So the pacemaker does exactly what it says. It sets the pace for your heart's beat. Most of us, our pace is naturally somewhere around 50. So if you had no brain telling your heart what to do, for most of us it beats about 50 times per minute. Now your brain is typically overriding that and your heart's beating like 60, 65, 70 per minute. If I start walking around faster, then I'm going to need more oxygen and more blood flow. So now my brain says, hey, raise his heart rate by 5 beats a minute. If I start running, it'll say all right, make his heart beat 90, 100 beats a minute, 110, 120. So my brain can override it. Here's what's cool though. My heart cells are joined to each other with a type of direct contact called gap junctions. Gap as in the gap. So gap junctions, which means these guys, that guy has a channel, he lines up with this guy that has a channel, which lines up with this one, which lines up with this one. So because of that, aren't they all sharing the same insides? So now what happens is this. That cell maybe leaks in some calcium. When he leaks in calcium and he contracts, that forces this guy to contract and this guy to contract and the next guy and the next guy and the next guy, and they all contract together at once. There's like a quick flow. Have you guys ever done the thing? When I was in 8th grade, I was in Miss Nichols' science class. And one day, she has us go into lab, and she's like, all right, everybody hold hands. And you're like, what's she going to do? So we all hold hands, and she's at the front of the line. We're all together just standing like this, holding hands. She's at the front of the line, and then she basically grabs this wire. So she grabs this wire, and the electricity just shoots through us. And you could see it as it went from one person to the next, to the next, to the next, to the next. And so times were different back then. Nowadays, she'd probably get a phone call for that. It's like, did you shock my son? It's like, yeah, but not much. Did you check medical records? Maybe. So she just sent a shock, and it just went from one person to the next. You could see each person go like that. That's what happens. This one contracts first. Because he's joined with his neighbor, they have the same chemical insides, the next guy contracts, and the next guy, and the next guy. And so it's just a ripple effect. So in your heart, pacemaker cells are just regular heart cells. But the pacemaker cells are the leakiest. So let me show you what I mean. We had a class of 20-something students, and let's say that all of y'all are heart cells. So all of y'all are just leaking in calcium. But over here, you guys leak in calcium like 50 per second. 55, 60, 65, 70, 75, 80, 85, 90. And then there's me. I leak in calcium 100 calciums per second. So I am the leakiest. Okay? So we say go. We start leaking in calcium. Who leaks in 100 calciums first? Me. In 1 second, I leak in 100. After 1 second, you guys have only leaked in 50. You guys have leaked in 60, 70, 80, 90, and then there's me that's 100. So I leak in 100 first. What happens whenever I leak in 100 calciums? I contract. But then I'm connected to all of you guys. So once I contract, then those of you close to me contract next. And then you guys, and you guys, and you guys. And so we contract from the bottom up, like that. Now, when we contract, we squirt out all of our calciums. Now, we're going to race again. Who wins the race for leaking in calcium first? Me. So, I make us all contract again. Everybody squirts out their calcium, including me. Now, we're going to let calcium leak in again. Who wins? Me. So, I'm the pacemaker. The pacemaker cells in your heart are just regular old heart cells. They don't look different. If you're dissecting a heart, they look exactly like all the others. They just happen to be the leakiest. So, whoever leaks in calcium first contracts first, and then since we're all connected with gap junctions, that sets the pace for the whole heart. You're like, all right, so now I've got a heart. Maybe it looks something like this. And my pacemaker cells are supposed to be like right about here. They send a message out to my atria, and then they send a message down to my ventricles. Kind of goes that way. Everything's cool. But what if my leakiest cells are not located right here? What if my leakiest cells are located right here? Now does my heart beat correctly? No. So now I need an artificial pacemaker. So then what we do, let's see. Give me 1 second. I got to pull up a picture. I'll show you guys a pacemaker. Give me 1 second. Grab that. Grab that. So many files, so many screens open. Okay, so I'll show you guys a pacemaker. I assume most of you all have not seen a pacemaker before. My wife has a pacemaker. Okay, so PICS, converted PICS, body systems, cardiovascular. Oh, that makes sense. Open it on that screen.
So theoretically, if you were like working out and when you work out your muscles contract, are you like putting calcium into your own muscle or is that just different? Like,
Yeah, you actually, do you remember the endoplasmic reticulum from biology? Like around the nucleus, there's this hollow ER.
a little bit.
You can like build proteins, package it in the ER, and then ship it. Well, we store calcium in our ER. And so what happens is whenever you shock a muscle, the ER squirts out calcium and your muscles contract. And then the calcium gets pulled back into the endoplasmic reticulum. So the way my muscles contract, like if I go like this, like that, the muscles in my forearm, what's happening is I'm shocking them, They leak calcium, my muscles contract, I suck all the calcium back, and then I shock them again, and I leak calcium. So your muscles leak calcium anytime electricity hits them. That's why if I stick my finger in a wall socket, all my muscles will leak calcium and contract, and I go like that. I don't know if that was a good enough answer for you. It's got a more complicated answer, but I think that might be good enough for the moment. So here's what a pacemaker looks like, and I'll switch cameras for you guys at home. So you got this little bitty pacemaker, and they put it just under your skin. So like if I had a pacemaker, you can see it right here in my shoulder, like right below your collarbone. And they stick it in there, and then there's little electrodes, little wires that they feed into your heart. I don't know how well you can see it on this x-ray, but there's a pacemaker, and then there's little wires that feed into your heart, and now the pacemaker shocks your heart for you. And so if this out of place, they call it an ectopic. Ectopic means out of place. If a woman has an ectopic pregnancy, that means the baby's like maybe stuck in her fallopian tubes or something. Ectopic means not in the right place. If I have an ectopic pacemaker, then the way around it is I just hook some wires into this pacemaker, and I shock it faster than your ectopic pacemaker, and so I now make this the new pacer. So I just have this little bitty thing under my collarbone with wires that feed into my heart, and it sets the pace for me. So it shocks me once a minute, let's say. Here's a lady with a pacemaker. The image is inverted, so that's why it looks like it's on her right side, but you know, the heart's over here. So pacemakers are always right here. And about every 8 to 10 years, it monitors its own battery life. So you can hook yourself up to this little thing, and it'll tell you how your battery life's doing. And about every 8 to 10 years, you go in and they cut it out, put in new batteries, and sew you back up. Bizarre. And just like everything else with surgery, somebody tried that the first time. Some surgeon's like, you know, if we had like this little box and it would just like shock your heart every second, we could like hook you up to that and it would shock your heart for you and it would beat for you. And other surgeons are like, let's try that. I bet it'll work. And then somebody did it and it worked. So my wife's heart, she has this thing called Wolff-Parkinson's-White syndrome, which is where her electrical pathway is messed up. So she had this alternate pathway that was basically the correct pathway is firing too slow. There's an incorrect pathway that's faster. So this pathway always wins. So when she was like 18 or something, they went in and they just tried to burn out this section, but they didn't get it all. So her heart at the moment without a pacemaker does not beat on its own very well at all. I remember one time before she had her pacemaker in, she was scheduled to get her pacemaker in. And we were sitting there, and she had on a heart monitor, and her heart just stopped for like 6 seconds. And she said, you know, it was weird. Like, I knew what was happening, and I knew that I was dying. She didn't die. Otherwise, why install a pacemaker? You know, it's a little joke. Okay. So basically, she's like, it was weird because, like, I didn't care. Like I just felt tired. You know how sometimes it's the end of the day and you just want to collapse and go to bed? Some of y'all might be like, that's every day. But pretend that it's like the roughest one that you've had. You're super tired. She's like, that's how I felt. It's like, I knew I was fading, but I just felt like so heavy and tired, like I was okay with it. And so I just remember sitting there like, I was holding her hand and everything, and she's like, she kind of gets this look, and all of a sudden I'm like, what's happening? And then later on, the episode was recorded on her little thing. They're like, yeah, your heart stopped for like, I can't remember what it was, 4 seconds, 6 seconds, something like that. So it was crazy. But now she has a pacemaker and everything's fine. So yeah, super weird. By the way, at some point, you know how they have like wireless charging on your phone? At some point, they'll be able to do that with pacemakers, so you won't have to always get a surgery to go in and get like new batteries. But right now, like, let's say I need to charge this thing for 8 years. It's not like you sort of put a wireless charger on there for 10 minutes. You know what I'm saying? Like, your phone, you put it on there for 10 or 20 minutes, and now your phone works for like several hours. This is like, oh, I need to charge this thing for how long? I don't know, like 8 years. You aren't just like 80%, 90%, I'm done. So I don't know exactly how it would work. But at some point they'll have that, which is pretty cool. So direct contact. An example is gap junctions. I have a channel lined up with a channel lined up with a channel. So this is like the best type of cell communication. We're literally sharing the same chemical insides. That's the easiest type of communication. The most fluid is I have gap junctions in here. And now what's happening in this cell is also happening in this one and this one and this one. That's the ultimate in cell communication. Okay, number 2 is paracrine signaling. I'll put paracrine down here. Paracrine is also cool. So paracrine is like a local signal nearby. Yeah, sure. So this is like a local signal. Let me give you kind of a video clip example of paracrine, and then I'll switch back so you can see the whiteboard. Let's see. Here, by the way, is like joining some heart cells together. These are heart cells joined together right here. You can see these little dark areas. That's where this heart cell, there's a heart cell here and under. There's a heart cell here and above, and they're joined right there. So they've got gap junctions, and they also have all these like protein fibers that kind of sew it together so your heart doesn't rip apart when it contracts. This is like 2 heart cells sewn together. My skin cells are sewn together the same way. You basically just take some protein string and sew it together. Pretty cool. Okay, now, let's say that I'm looking at paracrine signaling. This is the example of like if I get a cut on my arm. So they start with an animation, and then they show it with real cells. I'm going to turn down the volume.
Fibroblasts grown in vitro in a culture dish form a confluent monolayer of cells. Cells in a monolayer are relatively static. Contact each other.
All right, so here's a bunch of cells on like a microscope slide. We scrape down the middle, simulating a cut, and then the cells start talking to each other. So these are real cells, and they're going to grow until they bump into each other. Once they bump into each other, they're supposed to stop growing. If they don't, then that means they're growing out of control. And what's cells growing out of control called? Cancer. So, have you ever heard of somebody that like they went to the doctor and they found a lump or a growth? They won't call it a tumor, but they'll say, Grandma has a lump, so they're going to do a biopsy and check and see if it's cancerous. Have you all ever heard something like that? So, if there's a lump, then that cell's growing out of control. It is cancer. But there's cancer that doesn't really matter much. They call that benign. And then there's cancer that can actually kill you. So basically what they'll do is this. They'll take cells. A biopsy is they just get a sample. So let's say they want a biopsy of my liver cells. They'll stick me with a needle and like suck out some liver cells. Then what they'll do is they'll grow them in a petri dish. And they'll see when the cells bump into each other, when they make a monolayer, a single layer, that they stop growing. If they stop growing, we say that tumor is benign. When it runs into its neighbor, it stops growing. But if when they bump into each other, all of a sudden it just keeps growing and makes this giant pile, then now you have cancer. That's what they're doing during the biopsy. So if I've got some skin cells and I kill all the ones in the middle, somehow the cells on the side have to like talk to each other and let each other know when they're done growing. So the way that it works is kind of like this. Crap, I'm not supposed to have students on there. Okay, I'll do this demo in front of you guys, and then I'll kind of do a makeshift demo in front of you guys. So it's sort of like this. Let's say that all of y'all are my skin cells. You're in a monolayer, a single layer, and then I get a scratch. So basically, everybody in this row, y'all just died. Sorry. You're irreplaceable. But now we're going to replace you. So the way that we replace you is, let's say, actually let's take out this row and this row. That way you guys don't get too cocky. So now, everybody here is dead in these middle 2 rows. So who's going to fill these seats? Like, where are the new cells going to come from in this row? Yeah, from you guys. So you guys have to somehow sense that this row is now empty, right? Over here, you guys have to now sense that this row is empty. So you're going to grow, start filling this row. You're going to grow and divide, start filling this row. And then, once the seats are full again, you guys have to somehow talk to each other to let each other know, like, I'm here, quit growing. Does that make sense? So the way that cells do that is they release chemicals. So you guys still won't be able to see this very well remotely, but I'll do a demo version of it for you. So it's sort of like this. Right now, I'm sitting in the middle of the class, and pretend, it's typical. There we go. And I just sit here, and I say very faintly, I'm here. So could you guys hear what I said? I said, I'm here. If you're far away, you can't hear it. So I'm just sending out a very local signal. So instead of my voice, though, I'm sending out a chemical that says, I'm here. So I send out a little bit of a chemical that basically says, I'm a cell and I'm here. You sense it, you sense it, you sense it, you sense it. Everybody around me senses it. Okay, now, let's say that I get killed. So I'm gone. So now that seat is empty. So now, if I'm Sue, then she's going to be like, she gets a signal from her right that says, I'm here. She gets a signal from her left that says, I'm here. Behind her, she hears nothing. So she's going to grow and divide backwards. She's going to grow and divide, and now she's going to fill this chair with me. Once I'm here, I'll say, I'm here. She gets the message, so now she knows to quit growing. That's how it's supposed to work. But in cancer, you're deaf. So in cancer, she can't hear it anymore. So she doesn't hear anybody on the right, so she grows to the right. She doesn't hear anybody on the left, she grows to the left. She doesn't hear anybody behind her, so she grows. And she listens. I don't hear anything, so I grow to my right, grow to my left, grow behind me. I don't hear anything. I grow to my right, grow to my left. I don't hear anything. And then cells start to pile up until it's a giant mass called a tumor. So the analogy was, for those of you guys that are remote, this is going to lose some of its effect. So it won't be as dramatic because I just died in front of my students. So let's say that all these X's represent cells. And right now, I send out a chemical. I don't actually have a voice as a cell, but I send out a chemical, and that guy receives it. That chemical says, I'm alive. And it's sent out to this person, and this person, and this person. So now this guy right here knows I don't need to grow to the left. There's already somebody there. All right? When this guy dies, now there's no more chemical signal going to his surroundings. So now between these 4 people, somebody is going to divide and fill in the gap. Once the gap is there, now he's sending out a chemical signal that says, I'm alive again. And that's how your cells are supposed to know when to quit growing. It'd be almost like I grow until I bump into somebody. I don't bump into anybody. I grow and divide again. Again. Oh, and then once I bump into somebody, that's how I know when to stop. So with paracrine signaling, it's a local signal. I get a cut in my arm right here. These cells nearby are talking to each other. Cells over here aren't getting the message. It's just a local nearby signal. So paracrine is for like wound healing or something local. You're sending out chemicals. These guys are receiving it, and now they react. What if I send out growth hormone and I just keep making growth hormone? Then this particular set of cells will just grow and grow and grow and grow and grow, and now I've got cancer. So paracrine signaling. The next one is called synaptic signaling. Oh wait, I almost forgot this one. I got to show you this one. This is pretty cool. So in this little video clip that you guys are going to see, I got to turn down the volume again. Turn it down to almost 0 pretty much. So in this video clip, you're going to see a bunch of red blood cells. That's a white blood cell, and he's chasing a bacteria. He's basically following the chemical scent. So a neutrophil is a type of white blood cell. And so this guy is giving off a chemical scent, much like if I'm walking down, a dog might be able to smell my scent like a bloodhound or whatever. And so that white blood cell is following the chemical scent of the bacteria. He's picking up on a paracrine signal, a local signal, and he's chasing it. That bacteria is not purposely moving. That's just due to temperature. So he's just bouncing around, kind of like atoms would be bouncing around with temperature. But like this is happening in you right now. Phagocytosis. Thank you. 1 bacteria down, 50 trillion to go. All right. The next one that we want to look at is neurotransmission. Neurotransmitters, or synaptic transmission. So synaptic transmission only happens between nerve cells. Grab my eraser. I'll turn this back up in case we need it. So with neurotransmitters, that's how nerve cells talk to other nerve cells. So like, here's a nerve cell. Here's another nerve cell. And they can send signals to each other, chemical signals, to talk to one another. So if I'm looking at, we'll look at the board first. The way that you have a thought is when nerve cells talk to other nerve cells. So here's a nerve cell, a neuron. He could talk to nerve cell number 1, 2, 3, 4, or 5. But he sends a chemical message to nerve cell number 5. So that means nerve cell number 1 and nerve cell number 5, basically I send electricity, chemical signal. Electricity, that's a thought. So when those nerve cells fire together, that's a thought. If I'm thinking of like the color red, that might be because nerve cell 1 and 5 and nerve cell number 7 all fire at the same time. So nerve cell 1 sends a message to 5, sends a message to 7, and I think of the color red. We don't really know how this works exactly. Like, we don't know where the, like if you can visualize something, we don't know how it is that like when nerve cell 1, 5, and 7 fire together, we don't know how that creates an image in my head. Like if I think of my wife's face, we don't really know where that screen is. But we know that if we shock this part of my brain and make nerve cell 1, 5, and 7 fire, I will think of my wife's face. So we know that's where I store my wife's face. But we don't know how, like, okay, but like a smell, like I swear I smell it. Well, if we shock this part of your brain, you'll smell chocolate chip cookies. It's not really there. Right? Every once in a while, you go to bed, and it's really, really quiet, and all of a sudden, out of nowhere, in your ears, bing. How many of y'all have had that happen? Tinnitus. You're listening to music too loud, most likely. Is that sound actually happening in your room? No, it's all in your head. What happened was some nerve cells misfired in your ear. Again, probably because you're listening to music too loud. And so when they misfire, your brain's like, here's the sound that makes. Bing. It's all in your head. Your brain's just making it up. I've got nerves that go from my hand to my brain. Anytime those nerves fire, my brain says, feel it in my hand. Well, if I relax my arm at a 90-degree angle, I find those 2 bones between my elbow. That's my ulnar nerve. Try hitting that. It's your funny bone. It's at the bottom of your humerus, your funny bone. But if you do that and hit it, you'll actually feel electricity in your hand. You'll feel like a wave of electricity all the way there. How many of y'all can feel that? Maybe you suck at hitting your elbow properly. So there's a bone, and there you have to totally relax your hand like this. There's a bone and a bone. Right in between is where your ulnar nerve should be. When you hit it, if you ever hit your funny bone and it does that thing and you're like, oh, you like drop your pen or whatever. You drop your Expo pen that you're walking around the house with. That's what's happening. So anytime that nerve fires, my brain says, feel it in your hand. How many of y'all could get it to do it? How many of y'all can like feel electricity in your hand? Okay, thank you. So whenever you do that, your brain says, it's in his hand. I just hit the nerve here, but my brain says, that's coming from the hand nerves. So you should feel it right here. I'm currently telling my brain, no, I'm hitting my elbow. My brain's like, I don't care. Feel it in your hand. So I feel it in my hand. One last little magic trick. If I close my eyes, don't close your eyes yet because then you can't see what I'm doing. But if I close my eyes and look up at the lights and I do this, be gentle so you don't blind yourself. But if I push on my eye, so try that. Push like right where your nose meets your eye, just gentle. How many of y'all see like a splash of color in the corner of your eye? You see like blue or orange or red or whatever? Yeah, maybe yellow. What did you just see? Pressure. And it was blue or yellow or red. So what happened? Well, this is my eye. There's an optic nerve that goes from my eye to my brain. Here's my brain. Here's my eye. Anytime that nerve cell fires, my brain says that's coming from the eye, so you should see colors or black and white. So when I go like this, I smash that optic nerve against the back of my orbit, my skull. And so it sends a message along there. And my brain goes, blue? And I see blue. It's not real. It's not really blue. That's pressure. Right? You go to the fridge, you pull out some like habanero super hot salsa. You touch it, it's refrigerator temperature. It's 37 degrees Fahrenheit. You stick your finger in it. It's cold. You touch it to your mouth and you're like, it's hot. No, it isn't. It's cold. You're just tricking your brain. You eat some gum. I can have some like mint gum that's in my car. It's 110 degrees. I stick the mint gum in my mouth. I take a breath that almost freezes my teeth. You're like, oh, that gum's cold. No, it's not. It's warm. It's been in your pocket. It's been on the dash of your car. You're tricking your brain. So anytime those nerve cells fire, my brain is going to say, that's cold. But it could just be mint, and I fool it. Anytime these other nerve cells fire, my brain is going to go, that's hot. But it might not be hot. It might be really, really cold salsa. But I trick my brain, and my brain's like, no, 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 it's hot. Feel it. And I can even tell my brain, thermometer, it's cold. And my brain's like, no, it's hot. So when nerve cells talk to other nerve cells, we call that synaptic transmission. It's basically just a special version of paracrine. So like paracrine is when cells talk to nearby other cells. If those nearby other cells happen to be nerve cells, we call it synaptic transmission. So number 3 was synaptic transmission, nerve cells. And the last one, the one that you guys will think is the most interesting as far as pictures go, is endocrine. This is when cells talk to far away cells. When cells talk to far away cells. So now it's not a skin cell right here talking to a skin cell right here, or a brain cell right here talking to a brain cell right here. Now it's my adrenal glands talking to my brain 2 feet away. Endocrine is when cells talk to far away cells, and the chemicals that do that, does anybody know what the chemicals are in the endocrine system? The chemicals in synaptic are called neurotransmitters. Yeah, hormones. Neurotransmitter. They transmit messages between neurons. Endocrine is hormones. So hormones you dump in your bloodstream and they go everywhere, all over your body. So for example, let's say that we look at growth hormones. Like this picture. That's the tallest man that has ever lived. He was alive around the Great Depression, so like 1920s, somewhere in there. That's his dad. If you go online and try to figure out how tall his dad is, they always just refer to his dad as an average-sized man. So I don't know how tall an average-sized American man was back then. I'd assume that slightly shorter maybe, due to vitamins and whatnot, and it was the Great Depression. So maybe he's 5'8", 5'9", and that's his son, Robert Wadlow. Robert Wadlow eventually reached 8'11". These ceilings are 9 feet tall. So if Robert Wadlow had any height to his hair at all, as he walked in here, his hair would be touching the ceiling. Right? I'm 6, I'm 5'11". Usually doorways are 7-ish. So that's 9. So like, if I was standing like this, I'd still be looking at Robert Wadlow. If I wanted to look him in the eye, it'd be about right there. Like what I see right now is what Robert Wadlow would see. Just strange. You're like, oh crap, I dropped my pen. You know? Okay, so a basketball rim, how high off the ground is a basketball rim? 10. How big is a basketball net? Probably a foot, maybe a little bit more. So if Robert Wadlow walked under a basketball goal, his head would be like going through the basketball net. His head wouldn't touch the rim. Now if he did this, I bet he could. But as he was walking under a basketball rim, his head would be dragging through the net. Why? He had a tumor on his pituitary gland. So his pituitary gland released growth hormone forever until he died at age 22. So he was always in a growth spurt. You might have a friend that 8th grade or something, you left for the summer, you came back in 9th grade and your friend grew like 4 or 5 inches or something like that. You're like, holy crap, Robert Wadlow always did that. Every 3 or 4 months, you'd come back and you're like, holy crap, how are you taller? It's amazing. And this is cells sending out chemicals, hormones, to faraway cells. So when cells literally share the same insides, that's direct contact. When they send out a local signal nearby, that's paracrine. If that local signal is between nerve cells, we call it synaptic. And if we send out a signal to way the heck over there, that's hormones. That's the endocrine system. Like Robert Wadlow. Pretty crazy. The one last thing that I wanted to show you was a picture of a thought. So if you're like, what's a thought look like under a microscope? That. This is a nerve cell. The purple little vesicles right there, that's neurotransmitter. And so this is a nerve cell sending a message to another nerve cell. Its last message. So this brain tissue is underneath a microscope right now, so this like mouse is no longer alive. So this message right here is this. Rex, you're a mouse? That's your last thought as you're being killed? Okay. Never mind. So that's what a thought looks like. Super weird. That's what's happening right now as you think about it. Okay. So next class, what have we got? Lab. So make sure you read the first few lines of like 1A and 1B so that as soon as you get here, you can get rolling. I'm going to take roll and then check in over here with my home crew. See how you guys are doing. Don't see anything in the chat. You guys doing okay? I'm assuming you are. 10. Man, there's like 20 of you guys. This class just keeps getting bigger and bigger. All right. Well, if you guys don't have any questions, I'm going to take roll and kick on some TCTV. For you guys to see TCTV, the best way is probably if you go to the Talon and they load it up day by day. So, but otherwise, I'll see you guys Thursday, and I will take you into lab with us. So on Thursday, it'll probably be a little bit of like you're kind of doing science, but then you might have like lag time for 10 or 15 minutes. You can work on math. Then I'll like get your attention, kind of do some science. As far as like trying to visually see an endocrine signal, you can't really see it. You'd have to test for it. So you know how like in our little labs that we were just doing, you couldn't see the starch, but I know that starch is present if when I add iodine, it turns colors. That's kind of how endocrine signals work. So if I went to the doctor and I got blood work done and they wanted to see like what were my testosterone levels or my estrogen levels or my thyroid hormone levels or whatever. Let's say that I'm sick and we can't figure it out. They basically are able to use like machines, I know this sounds fake, but like mass spectroscopy and other things to identify chemicals. But you and I can't see it. We just have to run a test that makes it to where we can see it. If I was drawing a picture, I draw a picture of a cell here and a picture of a cell far away and then like a dotted line. And that dotted line would say like hormone above it or something. But you and I can't see it because it's like molecules moving, if that kind of helps. Any others? That was a good question. All right. You guys are free to go. I'll see you guys Thursday for lab. Bye. Yes. Yes. So when would you like to do it?
I think I have second period off tomorrow.
Okay, second. So I will need to put it together. Let me write myself a note. So the retest is 2 fresh free response. So there's no multiple choice, and it's not that I'm trying to be mean. It's just I don't have like another 30 decent multiple choice. If I wrote some new fresh multiple choice, they'd all be garbage. They'd be asking like obscure stuff because I've asked you all the good stuff that I've got. So it's 2 fresh free response. It might actually be, it'd be the equivalent of 2 fresh free response. So I might have it split into like 3 or something, but it would be the equivalent of like 2 10-point free responses. But yeah, I'll have it ready in Canvas tomorrow. You're welcome.
Can I ask what I should
Anything Unit 1 is fair game. So it is still the most important stuff. So if you think back to Unit 1, the big topics were like water and lipids and proteins and stuff like that. So it's still those. So whenever you took your test, the multiple choice and free response, if there were things that seemed to be the most important, there'll be different questions, but it's still going to be kind of the most important stuff, like carbohydrates. Carbohydrates weren't on your test a whole lot. So carbohydrates might be on here, but it's not going to be like the main focus or something like that. So anything Unit 1 is fair game, but most of it will be on like the big topics.
Thank you.
You're welcome. Happy to help. See you, Farrell.